It is my honor and privilege to introduce my new friend, Andrew Hessel. Andrew's title is Director of the BioNano Research Group at Autodesk, where he has the additional title, pardon me for all the titles after we just talked, the additional title of Distinguished Researcher. And I'm going to let him talk about himself, but I wanted to share with you just one thing that he told me last night when we met, which is that he feels empathy for molecules. And I know he will explain just what that means. And it's that type of thinking that caused him to be our choice this year for this audience, where we are all trying to make the transition in our mind from pure computer concepts to what, as Mike has named his new company, biomimetic concepts. And it's a transition that is long overdue, and one that I'm sure Andrew has a lot to say about. So please, let's give a warm welcome to Andrew Hessel.
It's a pleasure to be here this morning, and I'll probably turn down the gain on that. Where do I want to start? First, I just want to give some thank yous, because when I first got the phone call from Brad, I really didn't know if I was the right person for this meeting. We had a wonderful conversation, and he really convinced me just how special this group is, and what an amazing, I feel very privileged to be here. Thank you. I also had some wonderful conversations with Jim Spear, John Petty, Mark Anderson, and Joel yesterday that really helped me just kind of shape this a little bit to the audience, to you, and I hope I hit the right marks. So I've had an odd background. My first love was anything mechanical and electrical. I took it all apart. Never, about 50% got back together. And then I fell in love with computers as a teenager. So I wasn't formally trained in them, but the whole transition from mainframe to personal was fantastic for me. But then I ended up shifting my focus to biology. And that's because I really it was mysterious. It was wonderful. We hadn't solved the big problems, the fundamental problems in biology yet. And so I studied cellular, molecular, and microbial biology with a focus on genetics. And what I loved was there was such a close association between what I had been picking up in computers and what I was learning in biology. It was a really good mix for me. Today, and from there I went into pharma, biopharma, where we were literally programming cells to make medicines. Had a wonderful run with that. And then today I'm with Autodesk. We make design software, as you probably know. I won't go into it. But it's a really interesting place to land as a genetic engineer. I think I was the first one they ever hired. So I'm going to tell you some stories today about what I see coming in the future in terms of cell biology, real viruses, not computer viruses, and how I hope it's relevant to what you're thinking about these days and what you're doing in your own work. Now, my world is really new to humanity. We've always had the stars, we've always had the heavens. As long as we've had eyes and looked up, we've realized there's a lot to the universe. But the whole microbial world really opened up only in the 1600s. We needed to make microscopes to even have an inkling that there was anything below the level of resolution of the human eye. So bacteria, cells, germ theory, these are all new things. Viruses were only discovered in the late 1890s. So really, most of molecular biology, most of cell biology really has only come about for humanity in the last 100 years. I usually want to give people a sense of the scale. You guys are probably more familiar with this than most, but this is stuff you can see with your eye, and very quickly you move into the microscopic, where you need some sort of magnification. Large cells. Human egg is a large cell, sperm a small cell. Then you start moving down into the bacteria and some of the subcellular organelles like the mitochondria. And then you get into the nanoscale. Things like viruses, things like the low-level subcellular systems, the ribosome, antibodies, etc. This is an alien world to most people. Most people don't have any concept in their mind of what this world is like. At this level, you need electron microscopes to see it. So it's a very artificial lens into this world. And I know from my conversations with people, I might as well be talking about alien planets when I bring up any of these machines. All life on this planet, all life is based on cells. I don't study multicellular system, I study single cells. In fact, I spent years studying E. coli and Salmonella. They kind of look like hot dogs. They're about 1 micrometer wide and about 5 micrometers long. And in ideal conditions, they reproduce very quickly. It's, as you probably are aware, it's the main bacterial component of your gut, but it's not the only bacterium. There are hundreds of different bacterial species that exist in your gut. There are a lot of bacteria on this planet. There's 10 million trillion microbes per human. This is really the system, the low-level biological network on our world. If you drill a hole as deep as you can drill on the planet, you'll find microbes. Clouds are incubators for microbes. At the center of every snowflake is a bacterium that's nucleated the ice crystals. It's remarkable, just the diversity of microbes on this planet, how resilient they are, and really the types of biochemistry they can do. Now, this is a typical cartoon that you might get in a textbook, a high school textbook, on the structure of a bacterial cell. It's not really that enlightening, is it? Does it tell you much about how it operates, how it works? No. I have an 1,800-page textbook on the molecular biology of the cell, the Bible, that I keep on my, on my desk. That is a start. It's more descriptive. It's got more references. It still doesn't tell you how everything fits together and works. To give you a little more sophistication, I like this book, The Machinery of Life by David Goodsell. He's an artist and a scientist, and he draws these wonderful hand-drawn pictures of different molecular structures, and E. coli is one of his favorites. And this is actually a molecular motor in the E. coli. It has a hair-like structure called a flagella that it whips around at very high RPM to swim, to propel like a little outboard motor. And you can see that the molecular motor really looks like a motor. There's a number of genes that have controls. It can actually reverse and go forward. There's a clutch unit on this thing. These are very sophisticated little creatures. And everything at this level is in motion. This is just computer software called CellPack. But you have to imagine everything at this scale of matter is constantly moving. And of course, these little critters grow and divide really quickly. This is just standard textbook stuff from the 1950s. But under ideal conditions, they'll double in about every 15 minutes. So you start off with one bacterial cell. In 24 hours, you have billions of them. It's really remarkable. If you take an E. coli and you squish it, like you stomped on it with your foot, its genome comes out. That's, I know, it's kind of awesome, isn't it? It's an electron micrograph. But that's about 4.5 million bits of genetic information. Really, it's not much more than a single photograph. on your phone. But it's enough to encode the entire operating system of this very old bacterium. It's been around for billions of years. That's really interesting. E. coli has 4,500 genes. Essentially, think of it as code libraries. Humans have about 20,000. So this thing has approximately a quarter of our genetic metabolism in some ways. But it's so compressed, 4.5 million bits. When I saw that picture, I saw this, because my first computer actually had a cassette tape drive. And I realized, I can go, that's just how biology records data. It's digital data too, because each bit of DNA code is a bit. The cool thing about biology is you learn how to program DNA, and you can program anything that's alive on this planet, because it's the universal programming language. It's low level, but it's universal. And this was one of the things that really made me switch out of computers into life science. I figure if I can master this one programming language, I can work on anything. Now, bacteria are kind of neat. They have their own chromosome, that big one I showed you, but they also have these little tiny extra chromosomal elements called plasmids. And bacteria are really free with exchange of these plasmids. If you heat a bacteria up just a few degrees Celsius, E. coli in particular, it's meant to be at 37 degrees. It's tuned for the mammalian metabolism. If you heat it up to 42 degrees Celsius, it freaks out. Its metabolism is not tuned for that temperature. That's why we run a fever if we have an infection. And it starts to die. And so it will open up gates and channels in its cell wall to receive new DNA at 42 degrees to see if there's any code floating around in its environment that it can pick up and express and hopefully allow it to survive. It's kind of neat. So E. coli this is one of the reasons why we work with E. coli. It's incredibly easy to exchange DNA and to put new code into it. So these plasmids are what most of the genetic engineers work with. In fact, the way I like to explain it to kids today is the bacterial cell is like your cell phone. The chromosome is the operating system, whether it's iOS or Android. And the plasmids are the apps. And depending on what apps you load into that cell, it can do very different things. No two cells are exactly alike. If you move up the evolutionary tree into more complex cells, this is Baker's yeast. It's pretty much the same game. This is Baker's yeast, freeze-fractured in an electron micrograph. The main thing here is you can see these circular structures, well-defined, like little orbs. That's the nucleus. That's what the main difference is between a bacterial cell and a yeast cell. The yeast cell has a true nucleus. That's why it's called eukaryotic. Our cells, you and me, are eukaryotic cells. Now you can see it has a more complex cartoon. You can see the nucleus, the nuclear envelope, a whole bunch of other structures in there, mitochondria to generate energy. It's not just a bag of chemical soup like the bacteria. It's organized like a city. It's got a seat of government, it's got waste removal, it's got energy manufacturing, it's got transport systems. So the eukaryotic cell is about 1 billion years more advanced than the bacterium. Bacterium is like a small town. This is like a city. Basically, all higher life is built on this eukaryotic architecture. This is a human cell. If you look in that box, you can see everything's in motion. There it's starting to divide. Did you see the chromosomes separate? The chromosomes actually condensed, divided into two. You get a mother and daughter cell. You can see, boom, you can see those strands like fingers. Those are the chromosomes in a human cell, organizing, condensing, and then dividing. But you see how much motion there is down at the cellular level. This is why cell biology is so fascinating. It's just complete dynamism. Our real lens into this world, because our microscopes aren't that good, is through DNA sequencing. DNA sequencing is a technology that's been advancing exponentially for as long as I've been alive. And it doesn't allow us to see all the molecular structures, of course, but it allows us to examine the code of these fascinating little cellular processors. And it's gotten extremely cheap to do it. As you know, the Human Genome Project was started in 1990. In 1990, we had not sequenced any organism on this planet other than a virus, some of the smallest genomes that exist. But the scientists that launched the Human Genome Project were kind of visionary. They realized we might as well do a moonshot. Like, they hadn't even sequenced a bacterium yet. And they wanted to do the 3 billion base pairs of genetic code that makes up a human being. But they were smart. They realized that we are human-centric. Most of us don't care about bacteria. Most of us don't care about any other organism other than ourselves and the other humans around us. We're not trained to think about life. So by setting the target of the human genome, which was extremely controversial in 1990, they created a pull for the technology to advance very quickly. They knew it was going to happen. Today, sequencing is virtually fully automatic. You can do a human genome with this type of machine in a day. What used to cost $100 million per genome is today $1,000, including full analysis, interpretation, and consulting. And these are just factories. They're churning out so much data right now that we're having trouble keeping up with just storing it, let alone analyzing it. This is the golden age of sequence, and it's only going to keep going because we've only sequenced a fraction of the organisms on this planet. We've only sequenced a few tens of thousands of humans, and yet guaranteed by the time this century is over, we'll have sequenced everything. Everything we can ever find, and will get sequenced probably continuously. The whole process of sequencing is really fun. It's easy to explain to people. You're just taking chemical bits of information and making them electronic. That's it. That's all this machinery does. And believe me, back in the early days of doing this, it was dangerous work. It was radioactive. You could spend a year getting 300 bits of information and damn near poison yourself doing it. So I've never seen a technology advance so quickly. Computing moved pretty fast, but believe me, DNA sequencing has broken every record. We have a really comprehensive map now, the wiring diagram of a cell. We know most of the essential metabolic pathways. This was figured out by the biochemists long ago, but we've continued to add information. This is actually a database called KEG, but each one of those dots is specific compounds. You can zoom in on the enzymes that do the reactions, et cetera. This is the highest level organization of that metabolism. We don't have a fully complete model, though, a digital model of a single cell, even the simplest cells. Stanford University published this model in 2012. It's the most sophisticated we have. It's not perfect because it's still pretty flat. There's a lot of formulas in it. It's not all molecular based. The cell that they're actually mapping here is mycoplasma, which was sequenced in 1995. It was one of the first 2 bacteria ever sequenced. So we've had this genome for a long time. And right now they're working with E. coli. I think it's to be published this year or maybe next year, depending on how their work goes. But this is not an inspiring model of, of a living organism. How do I show a kid this and inspire them? But it speaks volumes to just how much work by how many people has gone into elucidating just this much about a single living cell. What I love is, now that we have this foundation in genomics, we can start to think about writing code. This is one of my favorite code editors. It's by a company called Benchling, started by a couple of young scientists. They just said, we want it all based through a web page. We want it very social. We want it very colorful and clean and graphical. And they basically got this product up and running in less than a year, and they've continued to add a lot of different institutions and researchers to it. This is essentially a word processor for DNA code. It's not very intelligent yet. It still requires, it basically displays the information, but you still have to know a lot of biology to be able to do any programming with this type of interface. But it's a big step forward to some of the desktop tools I used to use. It's free. There's a few add-on services. But it's a powerful system, and it is clean. So I've got electronic bits I can manipulate now. Doesn't do me much good, though, if I'm sitting in front of a computer and I want to understand how something actually works, though. So it's really important to take those electronic bits of DNA code and have some way to turn them into the chemical bits of DNA so I can pop them into my cells. Again, it's a pretty easy job with bacteria. And see how the programs run. Basically, compiling the code. This is what I use today. Companies like Gen9 Bio, based out of Boston. It's not the only company that does this, but they essentially take electronic bits and turn them into chemical bits. This is a DNA synthesis company. It's basically a printer for DNA. It's not a printer I can have on my desk today. It's more like a Kinko's. You know, remember when you needed color prints? You used to have to go to Kinko's and they had that big giant, machine sitting in the corner. That's kind of what it's like. It takes a whole company right now to compile genetic code. But they're getting better and faster all the time. This didn't exist when I went into biology. If you wanted to manipulate DNA code, you had to physically manipulate the molecule, cut it with special enzymes, and stitch it together with other enzymes in a tube, and then try and figure out, did the molecules stick together? Every time I wrote any code, it was an experiment. This just made it digital. So this DNA synthesis, gene synthesis, eventually genome synthesis, is really the most powerful technology that's come into the world and become widely accessible in the last 15 years. It's made genetic engineering software engineering. We don't have really great code editors. We don't have great compilers. But the workflow is identical. Hello, living world. I want to make this point right now. If you've ever written a line of code, if you know anything about computers, anything at all, if you know just hello world, you are qualified today to come and program bacteria. We have 8-year-old kids now coming into the field, learning how to use this path, these editors and these printers, and to put code into bacteria. Normally, these kids just make the bacteria glow. Making something glow is the training wheels of genetic engineering. But it's remarkable how kids, just like they picked up computers, love programming bacteria. And seriously, if this is something you want to do, I will shepherd your way into this world. There's one big limitation for me today, and it's this. To make anything complex, you have to write a lot of code. Right now, the technical ability, the routine technical ability of DNA synthesis is in this range, basically from 0 to about 50,000 base pairs of code, which is remarkable. I just want to make that clear. 50,000 base pairs of code is a stunning achievement, and we haven't fully explored that play space yet. But depending on the organisms you work with as a biologist, you might not have enough code to really do what you want to do. Plus, it's expensive. Those gene synthesis companies today charge anywhere from 3 to 10 cents a base pair. Most of them around 10 cents for routine work. So I don't know many people that want to spend $500,000 making an E. coli bacterium. It's just, even if you could stitch it together. A tweet would cost $14, right? Like this is not scalable in many ways. Most of the folks working in that green layer today have big grants, or they're building an organism that does something important that they have a business model for. It's not a play space yet. This yellow zone is where we're going. This is the state of the art right now. We can get up to approximately 12 million base pairs. Right now, there is an international genome engineering project to make a synthetic yeast. Yeast has 12 million base pairs of code, and it is taken 5 years for an international team to get close. They'll finish this year or next year. Only one group in the world has made a synthetic bacterial genome, and that is Craig Venter's group in San Diego. And this is the future. 10 million base pairs and above. When you can start making whole chromosomes, the smallest human chromosome is 50 million base pairs, and we have 23 of them, so they only go up in size from there. You can get to the 3 billion base pairs you need for a human genome. And then some plant genomes are just absolutely massive. They pad their genome for extra protection. But I know this. DNA sequencing dropped in cost faster than any technology on the planet. And I bet that DNA synthesis is going to follow that path too. Just a couple weeks ago, a really important paper was published in Science. Craig Venter's group published the design and synthesis of a minimal bacterial genome. This is the smallest genome that can sustain reproduction, bacterial cell reproduction. Craig had been working on this, his team had been working on it for about 20 years. This is a fully synthetic genome. They did a lot of engineering to first minimize the genome, but then to optimize it. You're engineers. Design, build, test. It's the same process you might use writing code or making anything else in the world. Just the tools are a little different, but it all starts with computational design. It goes to genome synthesis, and then ultimately booting and testing in the bacteria. And the picture on the right is just their assembly structure. We actually can only synthesize very short pieces of DNA, and we have to string them together in scaffolds. It's complex. There's still a lot of biology you have to do to write those genomes. And this is what it looks like. It's 473 genes. Just think of them as code libraries. 473. About 500,000 base pairs of code. I think it's around 543,000 base pairs of code. It's kind of a weird little critter. All those are cells that are essentially, it's a little ball of growing bacterial cells. But this is the important point, my take home. 31% of the genes in that genome, they don't know what it does. They have no clue what the biological function is. They know it's absolutely essential. If they take it out, organism won't grow. But they don't know what the function is. This is why it's so hard to program biology right now. This is why it's such a wonderful play space. It's because, seriously, we're just getting started. The yeast project is a little more advanced, again, because yeast are about 1 billion years more advanced than bacteria. So I just showed you the minimal genome. This is the maximum one we're working on these days. And these are some of the groups around the world that are synthesizing the 16 chromosomes in yeast. It's about 12 million base pairs of code. China's doing a lot of the heavy lifting. I have to say, the US government is really freaked out about China, but when it comes to genomics, they've been carrying their weight and doing the hard work side by side with scientists in an international community. If you ever get a chance to go to China's Genome Center in Shenzhen, go. If you ever have the opportunity to play with some of these technologies with the Chinese, go, because they're really interesting. Now, my group at Autodesk is only 20 people. Half of them are software engineers, half of them are scientists. I don't have the resources of Craig Venter. I certainly don't have the resources of the scientific community. So I had to be really strategic in kind of choosing what I wanted to work on there. So I focused on the dark area of synthetic biology, which were viruses. The world of synthetic biology has been growing rapidly over the last 10 years, but viruses scare people, and it's hard to get grants for them, and a lot of people just didn't want the negative optics. But I'm the type of guy that if I see a dark area, I gotta go shine a flashlight on it. And I'm also, a microbiologist. I know that viruses are essential in the world. Now, in the bacterial world, you might see a virus like this. This is called a T phage. They come in different flavors, but the reason why this is so visually compelling is it looks like a little hypodermic needle. And in fact, it operates like a little hypodermic needle. It will seek out the E. coli cell wall attached to receptors that it recognizes with its tail fibers onto the cell wall, and then it will inject the DNA in the head of that virus, what they call the capsid, into the bacterium. And it does this very effectively. And that little bit of code that is injected into the bacterium is so powerful, it can take over the operating system of that bacterium in literally a minute and tell it, go and make viral particles. And it will make viral particles so fast and so plentiful, the cell will explode. They'll literally pop. It will release thousands of more viruses. That's why they're called bacteriophage. They eat bacteria. Here's a little number that you can throw in your head. 10 million times more viruses on Earth than there are stars in the universe. We have no concept of these numbers. They're just fantastic, just magical fairy dust. But here's the thing I want you to know. All viruses are basically USB sticks. That's it. They're inert. They don't do anything. Without a cell to plug into, they don't do anything. You can spread them on toast, eat them for breakfast. Even Ebola, doesn't matter. It has to have a permissive cell, a processor it can run on. But I love them because they're really small. You don't need a big lab. They've got really small genomes. They don't cost a lot to synthesize. And they're non-metabolizing. They don't do anything if you're just trying to design them. They're also significantly under-researched. It's just one of those dark areas that you can go make a big difference in. This made them ideal for CAD/CAM. And thankfully, Autodesk wasn't freaked out by it. So we have, the foundation of our work in the BioNano group is to make this stuff visible, because all this stuff is invisible. So we took one of our viewer products for large models in architecture, and we rebuilt it to do large molecules. And I've got a little video. I hope it plays nice. So I did this actually on a hotel Wi-Fi. It's all served up on a web page. This is a segment of the virus. It's basically a jigsaw puzzle that self-assembles. You can see I've got pretty much, I can zoom in and out. I've got atomic resolution on this. I could go right down to the individual atoms. I can, right here, I'm doing the assembly. I'm saying put the whole viral assembly together from the components. Boom. Now you've got the full virus. Inside, there's a cavity where DNA will actually hide. I can explode the model if I want to go play with it. We're putting in all of the manipulators now. But this was the heart. Just getting this software to work for molecules was a big step. We got to show it off last week because the Zika model was published in Science, but it crashed most of the viewers that were available. From the viewer, you can hit print and make a 3D printed virus. People actually ask me, is that a real virus? It's like, will it hurt me? If I wing it at you, yes. But that's a $1,000 blob of plastic. Anyway, it's not hollow. You have to work on that algorithm. But I can also send that file to a DNA synthesis company. And what I get back, you see on the right there, is from Gen9. That's just a plate containing viral DNA. And if I take that viral DNA and I just add it to some bacterial cells and heat shock the bacteria so they pick it up, then you get something like this. That's just the lab bench doing the work. Oh, you can use robotic labs as well. This is really fun. This is the first cloud lab, by the way. You can all access it now. It's called Transcriptic. The CEO is like 26 years old. He's brilliant. This is a full-on molecular biology lab that you can access through an API. It's really awesome. So I tried all these different techniques for booting up the virus. And this is all you get. You get a growth plate covered with bacterial cells. You can't see them here because it's just the background. But wherever you have a spot there, that's where a single virus was picked up by, virus DNA was picked up by the bacteria, started making more viruses, spreading the infection, so to speak, and clearing some of the bacterial cells. This is called a plaque assay. And each spot you see there is a synthetic virus that's booted up. Now remember, this was a virus that started its design cycle as a file on my laptop. It did not ever, there's no biological starting point. I didn't have to go to a freezer and pull out a tube. It started as a file on my laptop, the genome was synthesized, and I dropped it on a living cell, and that living cell started to make virus particles, which I detected by its killing living cells. This is essentially viral engineering 101. Craig Venter was the first to make this particular virus. It's called Phi-X174. It really doesn't matter. It's just a safe virus to work with because it only infects bacteria. You can also use DNA as a structural molecule. I won't go into this, but we also have design tools for bending DNA. So you don't have to use it as a coding material. You can use it as a physical material. And as an exercise, my nano guys designed the Autodesk logo, and then we synthesized the genome, and it self-assembles into the Autodesk logo at the nanoscale. You can see at 100 nanometer resolution, you get a lot of logos. So, so there's probably more Autodesk logos in the world than there are atoms. No, I'm just kidding. But here's the thing that really made me so excited about building this platform for viral engineering. I've been trying to build this platform to make it digital for about 10 years because I saw it coming, and I also saw how it could be used. Right now, we make medicines for people in a mass market way, one size fits all, using the old Henry Ford model. It doesn't really work that well for things like cancer. Having viral engineering, this type of programming allows you to make personalized medicines, just like you can personalize anything with 3D printing. This is really cool because viruses don't cost much. How much did your last cold cost you, right? Small genomes, really cheap synthesis, virtually nothing in manufacturing. You can scale it to the world. In fact, you don't want to do that by accident. But most people don't know that some of the most advanced cancer therapies in the world today are based on viruses. Two types. You either use the virus to give cancer cells a cold while leaving normal cells alone, so they basically sneeze on each other and they all die. Or you use a virus that can train your immune system to go hunt down the cancer cell. This is called an immunotherapy. So you're basically making a cancer vaccine. The cost is about the same when you have a digital platform for designing viruses. So this is a game-changing technology for treating cancer when we get more people piling on and using these tools. So that's basically what I'm trying to do. I've gone to virtually every drug company in the world and said, you know, you've all been using the blockbuster model to make your drugs. That's going away. It's going to the Netflix model. One low subscription price, all the medicines you want, just for you. This is coming as surely as Netflix saw the end of Blockbuster. It's coming to pharma. It won't work for every medicine, but for cancer, it makes the most sense. And certainly as we learn more about our own personal physiology, it will make more sense for more diseases. Right now, I'm really fortunate that I'm working with this guy, Dr. Bruce Smith at Auburn University. He's a veterinary oncologist. He treats cancer in dogs and other animals, but he's also a molecular biologist. The guy's a unicorn. There's not many folks with those qualifications. And we've been working now to build the first synthetic, fully synthetic, designed starting with a file on my computer, fully synthetic cancer-fighting virus for dogs. Getting a cancer-fighting virus into the clinic for dogs is easy compared to making a cancer-fighting virus for humans. Incredibly easy. So I hope that by the end of the summer, we'll have treated the first dog. After that, that's pretty much all the work I'll ever do with live viruses, because now I'll have to create another company. Perhaps you want to be a part of it to accelerate that cycle. Autodesk is not in the business of curing cancer, but the tools will be accessible and we'll know how to use them. And it's really, once you start cranking on this system, we'll treat 10, 100, 1,000 dogs faster than any other clinical trial going on in humans in the world. We'll learn more about cancer by working in dog cancer and cat cancer than really human trials can proceed. Dogs really are man's best friend when it comes to clinical trials. We treat them as members of the family. We are absolutely going to be using animals more in the future to open these channels for R&D, to make it real, not just a paper, but so you can see results. So what we're seeing here is the coevolution, the fusion of silicon and carbon. You can't do this work without silicon. You need a foundation of silicon computing to do this work, even just to understand it. But programming carbon is so much more fun because it's real world, and it's all self-assembles. So what we're seeing here is an absolutely fundamental shift in the architecture of molecular biology, of R&D, of pharma, material science, fuels, take your pick, because last time I checked, life is kind of universal in everything that we do in the world. You know, if it's got a human involved, it's important. It is a massive opportunity space. Massive. Like, you guys saw the birth of computing. You've helped nurture it and grow it. You know that we've only scratched the surface of what computing can do. Life has populated this world with some awesome designs, but now we've got this opportunity to go under the hood and start really tuning them for human needs. This is incredibly important. Companies have started to move into the space. They started moving in about 10 years ago en masse, calling themselves synthetic biology companies. Craig Venter's Synthetic Genomics being one of the first. But you've probably heard of CRISPR. It's in the news a lot these days. It's just genetic cut and paste. That's all it is. Editas, one of the first companies in the space, that's Dr. George Church from Harvard, it's already worth $1 billion. I have no idea how much it's worth. They just showed that they can edit out the HIV virus from human cells with one quick and easy reaction. That's just, one of the first experiments. I have no idea how big this is, but genetic cut and paste is going to be massive, because now you can do genetic surgery. But you don't have to be a Harvard professor or Craig Venter. You can be like my friend Anthony Evans. He started a little company just to make a glowing plant. He put it on Kickstarter. He raised $500,000. He uses the robotic laboratories that I use. And now he's got a company called Taxa where he'll just do it as a service. You want a plant to make something? Talk to Anthony. He'll design it for you. He'll help build it. For 10 years, I've also worked with a program called iGEM, spun out of MIT. Tom Knight, someone you may know from the computing world, decided he was going to reboot his career and start programming biological systems. It turned into a program. It's now spun out of MIT. It's an independent foundation. iGEM stands for International Genetically Engineered Machines. It's pretty much the US first of genetic engineering. This was the 2015 class from around the world. I think there's something like 50 countries represented now, about 3,000 students a year. There's high school leagues. There'll probably be junior high leagues appearing. It is so much fun. You have to go to Boston at the end of October, beginning of November, just to get a sense of the energy of this place. Like, it is remarkable. Everyone here is basically under 25, and they're doing some of the most important genetic engineering in the world and doing it well. And the ones that are more serious are going to places like IndieBio. This is one of the first incubators, accelerators for synthetic biology, and for just biological engineering. You have an idea, you go to IndieBio, it's a really easy application process. They'll give you up to $250,000 in cash, in lab space, in mentorship. They've already, in the last 2 years, they've incubated over 42 companies. Some of them are going to be worth billions. These guys are smart. But I have to say this. Hacking biology, playing with biology, programming it, engineering it, is so unfamiliar to us today that as this stuff gets faster and cheaper, it's going to get weirder. So you have to have an open mind. If you want to run from this room now because of my talk, you're going to have to go a long way away from the world to not be touched by this technology. It's coming that fast and that hard. Here's some of the stuff I know is coming. Beer is the first biotechnology that humanity created. We started making beer 10,000 years ago. And I think it's the number 3 liquid consumed in the planet. There's like water and tea, and then there's beer. Anyway, as you know, beer is made by yeast. Yeast that we can now synthetically engineer the entire genome of. And yeast is eukaryotic, like you and me. And I'm running a little late here, but it's like you and me. So you can put any gene pathway, any metabolic pathway from virtually any creature into yeast. The students at iGEM were hacking this years ago, and they were making yeast that produced cancer medicines, new flavors. My favorite one was they put in the metabolic pathway for caffeine. So they made caffeinated beer. It was awesome. Like, this is really cool stuff. And I mean, like, this is accessible to everyone. And a couple years ago, I was mentoring a student in one of the earlier versions of IndieBio. And the student said, I want to port all of the cannabinoids from marijuana into yeast. And I said, I'm going to invest in that, thank you. Stay tuned, we're going to get some awesome beers in the future. This was published just a few weeks ago. This is an amazing study. These mice are the same age. The mouse on the left looks pretty crappy. The mouse on the right looks pretty healthy. It's because they've put a program in the mouse on the right that when its cells starts to senesce, just get old, they commit suicide. And of course, we always replenish cells. So if you just knock off the ones that are starting to age as they age one at a time, they get replaced with a younger, healthier cell. This does extend healthy living and lifespan in mice. And watch, it will come to dogs. a lot of things are going to come to dogs soon because we've got to try this out. Humans are going to want this program if it works. Nothing has to go extinct anymore. There are de-extinction programs going on in the world now. We're doing passenger pigeon, but woolly mammoth is on the list. I vote for Elvis. And the way we have babies is changing too. This is IVF. That micropipette holds a single sperm. The technician brings the sperm to the tip, punctures the egg, pulls out a little bit of cytoplasm, mixes it with the sperm, squirts it in, and that's how life starts. I know, because this is my daughter, and she was made by that process. And my contribution was in a little room with some magazines and some old DVDs. And they were pretty stained. I'm glad I had my phone. Let's put it that way. I hope you all have a chance to meet my daughter. Her name is Rosalind. We named her after Rosalind Franklin, the genetic scientist that produced the X-ray crystallography data that Watson and Crick used to decipher the double helix. She was one of the most forward-thinking and amazing women scientists on the planet. Anyway, she's heard every conversation and every story that I've ever told, so I don't know who she's going to end up being in life, or where she'll go, but it'll be kind of fun. I know in the next few years we're going to get an Eldon Tyrell, if you remember the movie Blade Runner, in his giant corporation in Los Angeles making synthetic humans, replicants, cyborgs. These technologies, silicon and carbon, are blending. It will be a while before we make humans, but we're going to see a lot of really amazing engineering, this interface between silicon and carbon happening for the next few years. This movie I saw, it came out in 1982. I love this movie. I also love J.F. Sebastian, the guy that lived in the decrepit building and had all his little toy friends. That's kind of the open source version. He's the industrialist. Choose your side. They still played chess together. They were still friends. Just be careful of what you wish for. And you don't have to take my word for any of this stuff. Alec Ross, who really advised Hillary Clinton as Secretary of State on innovation, just published this book recently, The Industries of the Future. Chapter 2, The Future of the Human Machine. The last trillion-dollar industry was built on a code of ones and zeros. The next will be built on our genetic code. And it's an amazing chapter. But this really is world-changing technology. I really want you to be a part of it. I want your kids to be a part of it. I want you to be vocal if you want it and vocal if you don't. I know that we have to rebuild basically the infrastructure of the world to manage these technologies properly. Today, if you get sick, if you get a virus, the way we detect it is you show up at the hospital sick. The doctors at your hospital do not have the tools to analyze viruses. They don't. They can't see them. They have barely any detection for it. Next generation sequencing, forget it. Everything has to change. If there's an outbreak, whether it's Zika or Ebola or the next flu, we're always playing catch up. That has to change. We have smoke detectors in every room. We need virus detectors in every room. We need virus detectors on your Apple Watch. There's no reason why we can't have it. These are the cheapest technologies on the planet. We've got to master them, or they will be used just to instill fear. Viruses are not our enemies. Out of the billions of viruses in the world, a couple of hundred that we know of cause human disease. And we know how to fight them, because we've got the same tools they do. So let's go build the future. Thanks.
So I've been really excited about this for a number of reasons. One of which is my first degree is molecular genetics before I began all of this. And we had one of the world's first gene sequencers that we could do 2 base pairs a day.
Wow.
That was back in
That's high throughput.
early 1970s. We have time for 2 questions. And then Andrew's going to be in one of the tech suite, in one of the briefing sessions after the tech suites. It's on your program, and he'll take detailed one-on-ones, detailed questions at that point in time. So let's start with Jay, and then Julian.
If I remember right, a couple of years ago at a symposium in New York, you talked about synthetic tissue development. Could you update us on that, particularly as it relates to bioprinting? And then secondly, when does Autodesk plan to have any kind of commercial technology that comes up out of your basement to bring into the market for molecular modeling and simulation?
Yeah, that's a great question. So bioprinting is actually at the crossover point into Autodesk's more familiar businesses. 3D printing you all know about, but you can actually 3D print living cells as ink, as some of the materials that you're depositing. We've been focusing our development either with partners like Epibone in New York that are 3D printing bone, or doing materials development in 3D printable substrates that are non-toxic to the cellular inks. We don't do the cellular engineering. That requires much more specialized biological expertise. But there's groups like the New York Stem Cell Center that are very good at that. I think you're going to start seeing applications come out in the next few years, but it'll come out of the hacker communities, I think, at the beginning. These are all pretty new. Epibone is one of the first to really have an interesting project that can go probably into the medical market quickly. So it's going to be noisy, and it's going to take a little while for the dust to settle. As for Autodesk, having product in this space, we're still part of research. There's no, we have no imperative today to sell any product, and that's because, look, there are so many competing software tools in this space that were written by academics or other groups. None of them are making much money. We just feel we have the time and the focus on the application space. We just want to build relationships and get feedback to make the tools better. This isn't a field that's going to be around for a year or two and then disappear. This is just going to get more powerful. We'd just like to start off on the right foot.
Okay, and Julian, stand up.
At one point you said genetic engineering is software engineering. And another point you said we are digitizing life. And to me, this almost says that you're turning all of this into a software revision problem. And if you can imagine now we have life as a GitHub repository, just think of the ramifications of all the pull requests. But more seriously, what I'm wondering about is, now that we have access to this entire history of all of the engineering that you're doing, how does this help with mutations?
That's an interesting question. First of all, it is like software engineering, but we don't understand the processor, so it's hard to program. And really, right now, the complexity of the software we're writing really is Hello World. So it's a long way to go. And it's Craig that's digitizing, says he's digitizing life. But in truth, all of biomedicine is digitizing parts of life and putting it into databases. It's just not consolidated yet. As for mutation, mutation is like disk corruption. If you solve disk corruption in computers, if data never corrupts, great. I don't think you've done that yet. Nature never solved disk corruption in biology. That's why it just starts with a single pristine cell and grows a new organism. That's one way to do it. When it comes to making this code, it's precise and pristine when it comes off the synthesizer. You verify it. But if you're making a living cell, it's always going to drift. That's why we're learning how to put in switches to give it short lifespans. Or that's why we can put in error correction. If things start to go wrong, it can shut itself off. But we're still playing with single-celled organisms. So there's still a long way to go in this. But mutation isn't as much of a worry as something like viral engineering in the wrong hands. We need new architectures to just be able to scan the world and do a better job of accelerating the positive applications of virology and not worrying so much about the fear.
Jillian, with respect to your question about mutations, 5 years ago, I believe, we had a Meiutic parataxis presentation from China on how to develop code for 100,000 years of successful error-free continuity at the gigabit level for E. coli, for data archiving.
So for data archiving, it's fine. In fact, you're seeing stories in the press all the time about this. They just digitized some movies, books were digitized. That works because you don't just write one version, one copy of the digital file. You write hundreds of millions of copies. So then you just assemble and do a consensus. You'll get an accurate copy of the code out of it. That's how biology does it in many ways.
With that, it's time for us to move over to the tech suites and the next stage. I can have another round of applause for Andrew.